Okay, hello to all of you. Let's discuss chapter number 19. And chapter number 19 is about the infectious diseases affecting the respiratory system. So let me go into slideshow mode. Okay, so in this topic, in this chapter, we are going to talk about some of the diseases of the respiratory system. The first one is pharyngitis. We'll talk about pharyngitis. Then we are going to talk about common cold, sinusitis, otitis media, diphtheria, influenza, and whooping cough, respiratory syncytial virus, tuberculosis, and in the end we are going to talk about pneumonia. So basically, these chapters there's a very straightforward information. You just need to know these things, so there's nothing to explain. So I'm going to go over some of the important things. The first disease is pharyngitis. Now pharyngitis is basically the inflammation of throat, and in this there is redness and the swelling in the mucosa, and there's a pain. And sore throats which are caused by bacteria, they're usually more painful as compared to the sore throats which are caused by the viruses, and they are accompanied by fever, headache, and nausea. So the viruses which cause pharyngitis are the same viruses which cause common cold. The most important cause of pharyngitis caused by bacteria is Streptococcus pyogenes, which is a group A streptococcus. Now, Streptococcus pyogenes. So this is the most common causative agent, most common bacterial causative agent of pharyngitis, Streptococcus pyogenes. So I'm going to give you a few characteristics of this bacteria. It's a gram-positive coccus that grows in chains. They do not form endospores, they're non-motile, they form capsules, they are facultative anaerobes, and they do not produce catalase. Okay. Now, the untreated streptococcal throat infections, they can result into some very serious complications like scarlet fever. So this is a result of an infection of Streptococcus pyogenes infected with a bacteriophage. Now what's a bacteriophage? Bacteriophages are the viruses which infect the bacteria. So scarlet fever is usually caused by this bacteria when it is when it is infected by a virus, and that virus we call a bacteriophage. And that bacteriophage basically is having genes for the production of erythrogenic toxin. And in this disease, in the scarlet fever, there is like a rash with high fever, and the fatality rate is up to 95%. And also streptococcal infections, they can also lead to rheumatic fever, and this rheumatic fever is due to immunological cross-reaction between streptococcal M proteins and the heart muscle proteins of the host. Okay. Now, why this bacteria is so virulent? Because this bacteria has some virulence factors. They have the ability of surface antigens to mimic the host proteins. So the bacteria has some antigens on the surface which can mimic the host proteins, and they also possess some super antigens. This bacteria also has a specialized polysaccharide, and this specialized polysaccharide protect the bacterium from being dissolved by lysozyme. So lysozyme is an enzyme. Okay, so this bacteria is covered by, or is having some polysaccharide coat on it, and which protects the bacteria from getting digested by this enzyme. Then they have lipoteichoic acid that contributes to the adherence of the cell wall to the epithelial cells of the pharynx. So this lipoteichoic acid, which is there in the cell wall of the bacteria, it contributes to the adherence, it helps in the adherence of the bacteria to the epithelial cells of the pharynx. Then they have M proteins which can resist phagocytosis. We studied in chapter number 12 what phagocytosis is. And then they have hyaluronic acid capsule which also contributes to the adhesiveness. So this is the structure of the Streptococcus group A Streptococcus bacteria. So you can see like these are the fimbres. Now fimbres we have studied in our preliminary chapters what fimbres are. So they have M proteins in these fimbres. Then they have some protein antigens on the surface. Then peptidoglycan is there in the cell wall. This is the cytoplasm. Then outside the cell wall they have a capsule, and in the capsule they have the hyaluronic acid. And this is the lipoteichoic acid. So this lipoteichoic acid helps in the adherence of the cell wall. So this is the cell wall which has peptidoglycan in it. It helps the adherence of the cell wall to the epithelial cells of the pharynx. So these are all virulence factors of the this bacteria, which is a very, very important cause of pharyngitis. Then streptolysins, they produce O and S, and these streptolysins, they basically cause beta hemolysis of sheep blood agar. So these things we have talked about in the lab, in the selective and the differential medium. In those, we talk about all these things. Then erythrogenic toxin. This bacteria also produces this toxin, which is responsible for the bright red rash, induces fever, and how this bacteria can be, how this infection can be diagnosed. So the swab samples can be taken, pharyngeal swab samples can be taken, and there's the usage of antibodies to detect group A streptococci is done. And also these pharyngeal swab specimens, they can also be cultured. They can be plated on sheep blood agar, and Streptococcus pyogenes causes the beta hemolysis. So if you go back and look there, you can in the blood agar and all that. So some microorganisms produces alpha hemolysis, alpha hemolysins, some produce beta hemolysins, and some produce gamma. So Streptococcus pyogenes causes beta hemolysis. Okay, prevention and treatment. No vaccine exists for them. Prevention is through good hand washing, especially after coughing, sneezing, and before preparing food and eating. Penicillin is the antibiotic of choice for this pharyngitis caused by Streptococcus pyogenes. Now the common cold is caused by over 200 different viruses, which include rhinoviruses, coronaviruses, adenoviruses, and respiratory syncytial viruses. Now the next disease is sinusitis. There are many causative agents, many viruses, various bacteria, fungus is less common. They can all cause the inflammation of the sinuses. So sinuses are the spaces in between. This sinusitis is the inflammation of these sinuses. And broad spectrum antibiotics are used for bacterial infections, and antifungal are used for the fungal infections. Next disease is acute otitis media. So this is basically a middle ear infection, and this is caused by Streptococcus pneumoniae, Haemophilus influenzae, and other bacteria and viruses. Next is diphtheria. Diphtheria is caused by a bacteria called Corynebacterium diphtheriae. It's basically a non-endospore forming gram-positive club-shaped bacteria. And the virulence factor of this bacteria is diphtheria exotoxin, which is encoded by a bacteriophage. So I have already explained what a bacteriophage is. Bacteriophage is a virus which infects the bacteria. And these phages, they have the genes for the toxins. So virulence factor for Corynebacterium diphtheriae is diphtheria exotoxin, and it is encoded by a bacteriophage. And this bacteria can be cultured on a medium called tellurite medium, and this bacteria forms gray and black colonies. And the prevention is by diphtheria toxoid vaccine. Next is influenza. So this is the virus which infects us every season, which we also called a flu virus. So for this virus, basically, this is the virus which is around very often, but still we do not have the vaccine for this virus. And every year new flu shots are made. The reason is this virus mutates a lot. So every season a new strain comes. So the vaccine which was, or the shot which was, a flu shot which was effective last year cannot be used this year because the virus has mutated, the virus has changed. So every year the new strain comes. So right now a lot of coronavirus is going on, but now in the fall there will be also influenza and corona. So there will be two viruses moving around, so more people are expected to get sick. And it will be difficult to tell who is infected with corona or who is infected with influenza. So in that case, the diagnostic tests will be very important. Okay, cyclic increase of influenza during winter months. So usually during winter months, this flu, influenza strains come around. Some strains may cause worldwide pandemics. So it was in 1800 and something when there was a big pandemic, which was because of this, which is also called Spanish flu. It was actually because of this influenza. But now we have overcome this virus, and now a lot of people are not dying because of that, because we have developed immunity and all that. So it's now a weakened virus. It's not that strong. And the symptoms of influenza virus are, it begins in upper respiratory tract, can progress to lower tract also if you don't take care, and or if it is going on for a long time, it can go to the lower respiratory tract also. So the symptoms are, headaches, chills, dry cough, body aches, fever, stuffy nose, sore throat, extreme fatigue, and it can also lead to pneumonia. So basically, influenza A, B, and C viruses, they fall in a family called Orthomyxoviridae. That's the name of the family. Okay, so they're spherical particles. They are 80 to 120 nanometers in diameter. They have lipoprotein envelope, which is studded with glycoprotein spikes. So they have lipoprotein envelope, which is studded with glycoprotein spikes, and they are of two kinds, hemagglutinin and the neuraminidase. Okay, so you can see here these ones are hemagglutinin H, and these ones are the neuraminidase, which is called the N. So these are glycoproteins, and they are studied in the lipoprotein envelope. And they have single-stranded RNA genome known for its extreme variability. As I said, it keeps changing. Okay? Single-stranded RNA genome. 10 genes on 8 separate RNA strands. Okay. So the virulence factors, basically this virus binds to the ciliated cells of the respiratory mucosa. Basically this virus causes severe inflammation, irritation in lungs due to cytokine storm. Glycoproteins and the structure are important virulence determinants. So these glycoproteins which we are talking about here, the hemagglutinin and neuraminidase, these are the glycoproteins, and they actually mediate the adhesion of the viruses to the host cells. Okay, and they also change to evade the immune recognition. So basically these glycoproteins, they change, and as a result of that, when they change, our immune system cannot attack them because they keep changing. Okay. So for the diagnosis of influenza virus, RT-PCR is a preferred method because culture takes long time. Vaccination, as I said, we get flu shots every year, new. But there are several types of influenza vaccines. They're all injectable. They can be trivalent, containing 3 viruses. They can be quadrivalent, containing 4 viruses. And these are some of the drugs which are there for the influenza virus. Amantadine and Rimentadine, they are basically used to treat and prevent influenza type A, and they do not work against influenza type B. Then we have Zenamivir. These are inhaled drugs, and they work against influenza A and B. Then we have Tamiflu. That's another antiviral drug. The next disease is whooping cough, or we call it pertussis, and the causative agent is Bordetella pertussis. And its incubation period is 3 to 21 days. It has basically a few stages. First is called catarrhal stage, which is characterized by running nose for 1 to 2 weeks. Then paroxysmal stage, where there are bouts of severe coughing. Then convalescent phase, where the number of bacteria decreasing and no longer cause ongoing symptoms. For the prevention, there is a DTaP vaccine, which is there for whooping cough. Vaccine does not provide lifelong immunity. Okay, the next is respiratory syncytial virus. So this virus basically causes the respiratory infection. So the transmission is through droplets in an indirect contact via fomites, and usually peak incidence in the winter and early spring. 100,000 children hospitalized each year in the US because of this virus. Virulence factor is syncytial formation. Okay. Next disease is tuberculosis, and this week there is a case study also on the tuberculosis. So tuberculosis basically infects our lungs, but it can go out of lungs also. So when tuberculosis is affecting our lungs, then it is called pulmonary tuberculosis. Okay. So 85% of the TB cases are contained in the lungs. So clinical tuberculosis can form primary, secondary. Secondary is the reactivation or reinfection, or the last one is extrapulmonary, when it goes outside the lungs. Okay, primary tuberculosis. Minimum infectious dose is about 10 bacterial cells. Bacteria multiply inside macrophages. Escape leads to cell-mediated attack on bacteria. There is the formation of tubercles in the lungs, and neutrophils release enzymes causing necrotic caseous lesions that heal by calcification. And the extrapulmonary tuberculosis, so it is that situation when the infection is outside the lungs. So it can be in the just give me 1 second. We need to stop this. I will continue from here.